All right, so hi everybody. This is lesson number 3, Industrial Biotechnology. This is going to be our first virtual lecture. I hope everybody's doing well and staying safe during our quarantine. I'm Miranda. I'm the project lead for the Columbia University Systems Biology Initiative, the outreach programs for high school students. I've been teaching for the beginning of this semester. Um, so you probably know me. Today I'm going to be teaching with Shayla. Shayla, you want to introduce yourself?
Hi guys, my name is Shayla. I am now currently working with Miranda in the committee for the high school workshops, and I'm excited to talk about industrial biotech.
Cool. All right, so first thing we're going to cover is what is industrial biotech? So industrial biotech is the exploitation of enzymes, microorganisms, and plants to produce commercial products. So industrial biotech aims to use renewable plant-derived carbohydrates, lipids, and other compounds to displace fossil fuels as the raw material and energy basis of modern industrial society. This shift is called the shift to a bio-based economy rather than a petroleum-based economy. So this development of industrial processes that are cost-effective and sustainable is working toward a bio-based rather than a petroleum-based economy. So a brief history of industrial biotech. When we think of industrial biotech, many of us think of it as a new field. And this answer is both somewhat correct and somewhat not. So we think of industrial biotech as complex artificially engineered organisms to make products of the future. However, it's really been around for a long time. Beginning in 6000 BC, industrial biotech was utilized for the first time as humans began to use the process of fermentation to produce wine and microbial yeast to produce beer. Another prominent product of early industrial biotech is cheese, which utilizes fermenting microorganisms to create cheese curds. This is what you can see on the right side of this slide. As you can see that cheese production, when you get down past standardization and filtration, you use fermenting organisms to create this step called coagulation or curdling. So during the 1970s and 80s, we begin to see the kind of industrial biotech that we think of today, where naturally occurring enzymes and microbes are used to replace the use of fuels and chemicals. So, for example, in this time period, many detergents used phosphates, and phosphates are a chemical that is really harmful when it gets into the environment. So at this point in time, there was somewhat of a crisis with phosphates from detergents leaking into our water supply. Since this was such a big issue, a lot of companies decided to replace their phosphates ingredients with enzymes. So enzymes are used in many natural-based detergents now. One of the ones that you might have heard of is 7th generation. So conventional detergents work by emulsifying substrates that are stuck on your clothes to remove dirt, but enzyme detergents degrade substances that contain fat, oil, or vegetable matter, and that's how they clean. Right, so common biotech practices. Basically, industrial biotech involves working with nature to optimize the existing biochemical pathways that can be used in manufacturing. The metabolic pathways of industrial microbes are exploited to yield commercially viable products. So the main powerhouse when we're talking about industrial biotech is something called microbial cell factories. So these cell factories are provided with a source of energy called a feedstock and are grown in large quantities to produce a high yield of a desired product. In general, there are 4 things that we want to take into consideration when designing a microbial cell factory to produce whatever product we're seeking. So first, there's the choice of feedstock, which is what we can see in part A of this diagram. So the feedstock will provide essential energy needed for your microbial organisms to live. The feedstock can be chosen based on availability, cost, and/or environmental concerns. In most cases that we have today, feedstock is called first-generation feedstock. So in this case, the industrial microbes get their energy from the sugars of edible plants, such as sugarcane and corn. So this is already a more sustainable alternative to petroleum-based production, or to the use of more endangered plant species. Focus is, however, on using something called a second-generation feedstock, and we haven't really gotten around to developing the technology to use this yet. But second-generation feedstock which is generally considered to be things such as cellulosic biomass that are straw, corn stover, forest residues, and other waste and residual biomasses that put less pressure on food production and cultivatable land area. So in this case, instead of growing things like sugar and corn that we can actually use for other things, and we are already having a hard time growing enough of it to feed our own population, instead of that, second generation feedstock aims to use waste that we already have a lot of and we already don't know what to do with in order to convert it into something that we have a use for. So the second thing to take into consideration is the importance of choosing the right microbe for the job. So there are many different microbial organisms that can be used as the platform for industrial biotech. The main ones that we tend to use are yeasts, bacteria, especially E. coli, and microalgae. So this choice of microbe, we call it the chassis organism. So this is the host microorganism that is used for the production of a desired product. So for the most part, most of the interest has been centered on yeasts, especially baker's yeast, that are already very well known and adapted for industrial conditions. The next thing is to design the environment for the microbes to live and grow. So you need to be able to have a large volume of these microbes living in the same place in order to get enough of a yield of your product to actually have a commercially viable production system. So different microbes need different types of environments to live and work well. So obviously one of the most important things to consider with this is how your microbe gets energy and how it metabolizes things. So obviously, if you're using microalgae, your microalgae need to be exposed to light because they use photosynthesis. If you're using yeast, you can have them in a dark environment. And you can have them also in a deoxygenated environment because they use an anaerobic process to live. So all of these things are important to consider when you're designing the place where your microorganisms will live, grow, and work. Another thing to consider with that is how much feedstock to give them and what other kinds of compounds you'll need to have wherever they're living so that they, while they're producing this product that they may not normally do, so that they don't die of toxicity from byproducts that are produced along the way. So the last thing to consider is that we must consider conversion pathways from the product of microbial cell factories into the desired final product. So in many cases, microbial cell factories, while very useful at producing large quantities of simple products, they can't create the more complex desired products themselves. So an essential part of the industrial biotech model is finding chemical conversion pathways of these readily available precursors that the microbial cell factories produce into their desired final products. And we'll take a look at that with a couple of examples later. So limitations to classic industrial biotech. We can obviously think of industrial biotech as a really amazing thing, and in many ways it is. However, there are still many limitations on the ways that we understand industrial biotech that place limitations on how much we can use it today. So the first is availability of sustainable feedstocks. When we talked about feedstocks, we talked about first generation versus second generation feedstocks. So once we get to the use of second generation feedstocks, those kinds of supplies are very sustainable. We have a lot of biomass that we don't necessarily use in our own products. However, right now, while we're using first generation feedstocks such as corn and sugar, there is production cost to factor into whether using the industrial biotechnology processes with that is going to be sustainable. The second is improving yields of product. So we can do a lot to our organisms to try and get as much product as possible out of these microbes. But the way that we have produced them now, it's very hard to get a large yield of a specific product out of these microbial cell factories. There are only a few things that we have learned how to improve natural yield enough to make a commercially viable supply. So that is another thing that we have to overcome. A next thing is improving understanding of metabolic pathways. So while we understand the pathways very well of some microorganisms, such as yeast and E. coli, There are still many, many other microorganisms that we don't understand enough of the complete genome and the metabolic pathways that they use them in microbial cell factories or as a part of industrial biotech production. So this is a limitation because these other organisms could potentially be capable of producing other products that we may want. But since we don't understand the pathways enough to use them, we're incapable potentially of producing those products. Um, there's also the concern of developing more robust microbial strains. So obviously, living and growing in these industrial environments puts a large strain on these microbes that we don't normally see in nature. So one of the concerns that is being addressed in research now is developing more hardy strains that are going to be able to live and work well under the stresses of industrial production. And then finally, there's ensuring that modified organisms cannot escape into the environment. So industrial biotechnology does not always use modified organisms. It can use perfectly natural ones. But a lot of the ways that we find industrial biotech to be useful is when we modify organisms to make the yield better or to make a different product that we want. So these modified organisms cannot be released into the environment because we don't really know what kind of damage they would do. It might be nothing, but you never know. So it's really important that we keep these when we're designing our industrial housing for these organisms. It's really important to make sure that they are isolated from the environment and to make sure that any waste products that may contain these modified organisms also is taken care of properly. All right.
Okay, so now that you've heard a very detailed introduction of industrial biotech, we're going to transition into the role of synthetic biology within industrial biotech. I'm going to start out by talking about what synthetic biology is and just give a brief summary in order to move forward and talk about its role in biotech. Synthetic biology is a fairly new multidisciplinary field that involves the redesigning of organisms for new purposes by engineering them to have new abilities. For example, synthetic biologists will identify gene sequences that they want to be expressed in a model organism and then give that model organism certain traits. They can create DNA chemically in the lab that encodes a gene in one organism and insert that DNA into model microorganisms such as yeast or E. coli, which naturally would not express this added gene. While looking at synthetic biology in combination with genetic engineering, there are two different approaches that engineering these microorganisms can follow. There's something called top-down engineering and bottom-up engineering. We'll start with top-down engineering as it's a little bit more commonly practiced and a bit more straightforward. Top-down engineering approach starts with a living organism and manipulates the cell's metabolic pathways and expression to produce a desired set of products. And for a top-down approach, you start with an unmodified or simplified cell and add foreign elements to it. For example, you can add new genes that encode proteins that synthesize a molecule of interest. A bottom-up approach involves a combination of natural modified and synthetic biomolecules as building blocks to essentially combine non-living components to create a living system that is designed specifically by scientists to yield a certain product. Bottom-up approaches are inherently more challenging than a top-down strategy, and in industrial biotech and many synthetic biology applications, top-down approaches are more common as well. So on the next slide, now I'm going to introduce the concept of metabolic engineering. A metabolic pathway is defined as a set of interactions between genes and their products that essentially lead to the formation or change in a part of a molecular system. Metabolic pathways are essential for proper functioning of biological systems in every living organism. And a common metabolic pathway that you may be familiar with is glycolysis, which is a part of cellular respiration. Glycolysis does glucose oxidation in order to obtain ATP, which is essentially providing energy for the cell. Metabolic pathways are a series of biochemical interactions to help convert molecules or substrates into usable byproducts. Yeast is a common model organism that is used in industrial biotech, as Miranda is talking about previously. Common metabolic pathways in yeast include cellular respiration and fermentation, which is used a lot in industrial biotech. Fermentation can produce a series of byproducts that can be determined and manipulated through genetic engineering. Later, we will talk about a few examples to kind of depict the role of yeast in industrial biotech. So metabolic engineering is directly modifying metabolic pathways in order to control microbial synthesis of desired products. And it's important to study the properties of integrated metabolic pathways and their genetic regulatory networks in order to be able to optimize their cellular processes. The optimization of cellular processes through metabolic engineering is extremely important in biotech. So recently, metabolic engineering techniques have been applied to enhance the production of industrial bio-based products. And these metabolically engineered cells can either overexpress controlling genes or key enzymes, but also block undesired metabolic pathways. To reduce a reliance on fossil input, metabolic engineering has been developed to produce environmentally friendly and cost-effective biochemicals from sustainable products and materials. So in the next slide, the slide has two images that show the steps and outcome of engineering metabolic pathways. So the basic goal is to redesign parts of a pathway by simultaneously engineering multiple steps in the pathway. This happens through a combination of many techniques, and the most common strategies I will talk about, they're simplified, a simplified version, but they are including increasing the metabolic rate or flux through the rate limiting steps in a pathway. They include increasing the supplies of precursors, which are chemical compounds preceding another in a metabolic pathway, by blocking branched chain pathways, which lead to byproduct formation, and lastly, removing feedback controls in the pathway. So I'm going to point out these steps now on the image on the left. So they are all observed in the figure on the left with the arrows. And between A, B, and C, we have a pathway engineered to increase rate limiting steps. C to F is blocking a competing pathway. And then after E is the redesigning of this metabolic pathway to remove feedback inhibition. And the yellow arrows towards A show increased supply of precursors. So these are all possible genetic manipulations to metabolic pathways in order to produce either an increased amount of a targeted product or a decreased amount of an unessential byproduct. And then the image on the right, just to simplify, shows that starting on the left, you are using a native E. coli cell to produce a lower yield of a product, but through metabolic engineering and modification of related pathways for whatever this target product is, you can lead the cell to produce a higher yield through engineering the E. coli, which is shown on the right of the image. So now I'm going to talk about how synthetic biology, which we first went over, and metabolic engineering, what we just talked about, and how the combination of these two fields is now the direction industrial biotech has taken. So just a quick recap, synthetic biology aims to design the libraries of genetic components, including promoter coding sequences, transcriptional factors, and more, and the assembly of genetic circuits that can be implemented in a model organism to produce a desired result that isn't natural to that organism. While metabolic engineering seeks for the optimization of cellular processes that are endemic to a specific organism to produce a maximized amount of compound of interest from a substrate that is in industrial biotech that is preferably cheap and simple. So both synthetic biology and metabolic engineering use databases and libraries of components and conditions to essentially generate the maximum production rate of a desired chemical compound. So overall, while synthetic biology provides the component information about different biological components, metabolic engineering tries to actually apply all this information to the optimization of biological synthesis of the desired compound. So the image on the right, having genetic libraries of organisms are extremely important in order to characterize different enzymes and genes that are important and play a large role in the metabolic pathways. In order to create and manipulate metabolic pathways in organisms and include synthetic genes to produce a new compound, it is important to have a deep understanding and databases of these genomic libraries in order to build and manipulate these cells. So just to summarize, metabolic engineering slowly improves the yield of naturally producing organisms. Synthetic biology is taking these microorganisms and adding non-natural gene elements into the organisms in order to produce a result and product that is not native to that organism. In a broader context, the combination of synthetic biology and metabolic engineering is about the engineering and manipulation of previously mentioned cell factories for the biological manufacturing of, just two examples, chemical and pharmaceutical products. So we'll now be going into some specifics to talk about applications of synbio and metabolic engineering within the industrial biotech field.
All right, so the first thing that we're going to talk about this is the commercial production of semi-synthetic artemisinin in yeast. So this is one of the first examples of using synthetic bio in conjunction with industrial biotech to produce more of a product. So first of all, artemisinin is a chemical that's used to produce anti-malarial drugs. So many drugs that we think of today are produced by pure chemical synthesis, but others, like in this case, artemisinin, are naturally occurring and may be harvested from natural organisms. So naturally, artemisinin is naturally produced by a plant called Artemisia annua. So its availability in nature is pretty limited because the plant takes a really long time for its growth cycle, and there's naturally always going to be fluctuations in crop yield. So therefore, the supply and price of the drug have fluctuated substantially over the past decade. So as you can see in this graphic on the left, you can see how long it takes for cultivation and extraction of artemisinin from this plant, A. annua, and it normally will take 14 to 18 months. So this causes a lot of fluctuation, obviously, because there's not always the same amount of demand for this anti-malarial drug. However, you always need to have some on hand because in case anything is going to arise. However, it's really hard to have this on hand because it's not super profitable for cultivation of this plant to occur all of the time. However, there's a lag in between when there's a diagnosed necessity for this chemical and when it can actually be obtained due to this long growth cycle. Today we're going to be talking about the production of semi-synthetic artemisinin in yeast. So this was researched by Chris J. Patton and J.D. Kiesling originally, and their aim was to provide an alternative source of artemisinin that could stabilize the supply and price of anti-malarial combination therapies, which are called ACTs for my purposes. So the way that they did this was that they engineered baker's yeast to produce artemisinic acid, which is a precursor of artemisinin, and developed a synthetic organic chemistry pathway to produce semi-synthetic artemisinin identical to the plant-derived compound. So we're going to go into this in more detail on the next slide, but for a brief overview, if you can direct your attention to the graphic on the right side, this provides a pretty good overview of what's happening. So you can see first on top, they talk about Artemisia annua, which is the plant where artemisinin is naturally produced. So there are 6 genes that are identified in the plant's genome that are needed for the production of artemisinic acid. And the DNA was sequenced and used to design new genes that are compatible with the yeast cell and optimized for efficiency. So on the other side, you can see in the blue, we have yeast. So researchers have identified a metabolic pathway in yeast that is able to make the starting material that's needed for artemisinic acid synthesis. Artemisinic acid is not artemisinin fully, but it is able to be converted to artemisinin in only one step rather than many. So it's a late stage precursor. So as you can see as we move down the graphic and into our tan yeast cell area, There's a new metabolic pathway that's designed. So anything that's in green is originating from the plant, and anything in blue originates from the yeast. So first you can see on the left upper side of this yeast cell, you can see the genome of the yeast cell, which now includes this new modified bit of DNA from the Artemisia annua plant. So as you move around the cycle of transcription translation, you can see where this modification starts to come into play, as the first stage is producing acetyl-CoA. And then you can, from acetyl-CoA, using only the genes that were naturally occurring in the yeast cell, you can encourage acetyl-CoA to produce FPPP. After this, yeast would not normally produce any more steps on the way to deriving artemisinin. However, since the introduction of these green Artemisia annua genes, you can now produce from FPP artemisinic acid. And this artemisinic acid, then once you move outside the cell, so that would be when it's secreted by the yeast cell, then chemical conversion can be used to turn this artemisinic acid into actual artemisinin. So in a more detailed look, if you want to start on the left hand of this slide, so the left hand of the PowerPoint slide shows the pathway for artemisinin production in A. annua. So this is how it's naturally produced. So this process works from photosynthesis to get energy, and a byproduct of photosynthesis is acetyl-CoA. So acetyl-CoA is then converted in a few different steps. So it takes a couple of different steps to produce FPPP, or FPP, which is an isopropanoid. So then after this, we're able to convert FPP to amorphodiene. And this is where things start to get a little bit more complex. From amorphodiene, there are two different pathways that the plant can take. It can either produce artemisinic acid, which is a precursor for artemisinin, or it can produce dihydroartemisinic acid, which then is converted through some mechanism with sunlight into artemisinin. So the plants themselves have different ratios of these two products, artemisinic acid and artemisinin, and it depends mainly on the strain of plant. For the purposes of designing a new pathway to get to artemisinin from a different origin, they decided to use the pathway from amorphodiene to artemisinic acid because the pathway from dihydroartemisinic acid to artemisinin is not well understood, but we can design pathways to turn regular artemisinic acid into artemisinin. So artemisinin is an isopropanoid molecule. So yeast is naturally able to produce isoprenoid because yeast has the mevanolate pathway. It indicates that it has the ability to produce the artemisinin precursor, artemisinic acid, because it has the ability to produce chemicals in the isoprenoid family. So in the middle diagram, we can see the pathway that Padden and Kiesling's yeast were engineered to have, which causes them to produce artemisinic acid. So the genes marked in yellow are part of the mevanolate pathway and are naturally found in yeast, while the genes in green are from A. annua, the plant, and are either inserted right into the yeast genome or incorporated on plasmids. So through the natural mevanolate pathway, we're able to get to the product FPP. So you can see that that is how we move naturally in the yeast from acetyl-CoA to FPP, because the only steps that we see before we get to the product FPP are yellow. So that means that they originated in the yeast. Once we get past FPP, however, there are the use of many green genes that we see, which are the ones that are from the plant and were inserted. So the genes ADS and CYP71AB1 are inserted to convert amorphodiene to artemisinic alcohol, and then ADH1 and ALDH1 are required for conversion to artemisinic acid. So as you can see on this diagram, there are two ways that the foreign genes have been incorporated. Some of them these little arrows that have these straight lines on either side, those have been incorporated straight into the yeast genome. They're completely integrated with all of the rest of the yeast genes and are completely part of this pathway that we see back here, which is on that big circle of DNA in the corner. That would be considered the yeast genome. However, there's also insertion on a plasmid, which we've talked about plasmids in the earlier lectures. It's just a small circle of DNA with only a few genes on it. So in this case, the plasmid is inserted with ADS and CYP71AB1. So plasmids and the bacterial genome work together to create this product. So from there, as you can see, after we use ALDH1 to convert artemisinic aldehyde into artemisinic acid, then chemical conversion is needed to produce the actual product that we desire, which is artemisinin. So the way that we collect artemisinic acid is that it's when these yeast cells are all grown together in vats, they're grown anaerobically. So they're in these dark vats that are full of liquid. They have feedstock, and in general, this product, artemisinic acid, is secreted by the yeast. So when it's secreted, it goes into the broth that these yeast are being grown in, which we tend to call the fermentation broth. So after this, we need to isolate the artemisinic acid from the fermentation broth. And then from there, a number of chemical processes are used. You have to use esterification to get to an intermediate form. You use something called the Hock process to get to your final form of artemisinin. We won't go into the details because it requires a lot of knowledge of organic chemistry. But in general, the thing to notice is that artemisinic acid is isolated from the fermentation broth, goes through a few synthetic chemical pathways, and then eventually is converted to what we wanted, which is artemisinin in its entirety. So this pathway was originally designed by Padden and Kiesling. Since then, the technology has been improved for higher yields and more efficient production, and two companies have taken the lead on this. The companies are called Amyris and Sanofi. So Amyris was the first one that really took off with production of this, using something called the high-rise technology, which allowed them to engineer these strains more quickly and allowed them to produce a better product yield. Sanofi got into the game a little bit later, and while it uses pretty much the same pathway, its main difference is that during the chemical conversion of artemisinic acid that's produced by the microbial cell factory, during its conversion to artemisinin, it harnesses the use of photosynthetic energy for this process. All right, so a second example that we're going to talk about with industrial biotech and synthetic biology is biofuels. So the search for renewable resources has become a global priority due to scarcity of petroleum-based fuels and the rising levels of atmospheric carbon. So isopropanol is a secondary alcohol that can be produced by microbes, and it can also be used as a biofuel to partially replace gasoline, among other uses. So there are bacterium of a strain called Clostridium that produce isopropanol at low concentrations naturally. So you might think that it would be useful for us to use these microbes in our industrial biotech setup as the chassis organism or the principal organism for our microbial cell factories. However, this is not really feasible for us because we don't know that much about this type of bacteria. Therefore, we sought to find a way to produce isopropanol in E. coli rather than trying to produce it in high quantities from Clostridium. So if you look at the left, graphic on the slide, you can see a pathway from glucose to isopropanol. So in general, this pathway is a modified pathway for E. coli. So this is the engineered pathway that researchers tried to use to get elements of Clostridium into the E. coli pathway in order to have E. coli produce isopropanol, which it normally wouldn't. So as you can see, the steps from glucose to pyruvate are unmodified. They're all natural in E. coli, but once you get to acetyl-CoA and afterwards, you have a lot of different choices. So I chose this example because when we were talking about the production of artemisinin, we talked about the genes that were changed, but we didn't talk about the selection process for which parts of the metabolic pathway would produce the best outcome. So in this one, you can actually see the choices that the engineers had to make in order to produce the highest amount of isopropanol yield from their E. coli. So once you get down to acetyl-CoA, the next step that produces acetoacetyl-CoA, you can see that there are two choices for the gene that enables this change to happen. So the first one is ATOB, which is found naturally in E. coli, which is indicated by the parentheses next to it, EC. And then the other gene that we can use is THL, which is naturally found in Clostridium A. So that's our first choice for which type of gene we want to use to get acetyl-CoA converted to acetoacetyl-CoA. After that, we have another choice. We have if we want to use the gene CTFAB, which is another one that's found in Clostridium A, or we can use ATOAD, which is found in E. coli. So that gene helps with the process of turning acetoacetyl-CoA into acetoacetate. After this, the next step has no choice. There's no naturally occurring pathway in E. coli that can produce the change from acetoacetate to acetone. So in this case, we're always going to be inserting ADC, which is from Clostridium A, into the acetone, or into the genome to produce acetone. After this, we have a last choice in how we would like to proceed from producing isopropanol from this acetone product. So there are two things that you can use for this. There is either ADH from Clostridium B, or there is ADH from another organism, which is indicated by the TB. So we're taking genes from a few different organisms for this. We're going to call them Clostridium A, Clostridium B, TB, and E. coli. So there are a few different choices that you can make. All of them will produce the end product of isopropanol. However, they can produce it in different rates and with different byproducts. So if you look at the right hand of the PowerPoint, you can see how each choice for which gene you want to use in this pathway affects the outcomes of what these E. coli are producing. So we're going to take a look at two of them. So we're going to take a look first at this second to last option, which produced by far the most isopropanol with the least amount of byproducts. So if you look at the chart, the way that you would read this is that they have the plasmids labeled. There are 5 different combination plasmids that they used. If we're looking at plasmid 4, you can then look through and see where the pluses are to see which particular genes were used for any given step in the pathway. So for this one, the plus is for THL from Clostridium A. So in that first choice in our pathway where we could use ATOB from E. coli or THL from Clostridium A to produce acetoacetyl-CoA from acetyl-CoA, we have chosen to use the gene that's produced in Clostridium A. As we move further down, we can read first that ATOD or ATOAD from E. coli was used for the second pathway. So instead of using the Clostridium A gene, we're using the natural E. coli gene for the step converting acetoacetyl-CoA into acetoacetate. After that, moving a little bit further, we can see that ADC from Clostridium A is used, which we knew that was going to happen because that's the only option we have. And then finally, we can see that for this last step, we have ADH from Clostridium B was used instead of ADH from TB. So in summary, the genes that we have decided to use are first Clostridium A, then we used a gene from E. coli, then we used another gene from Clostridium A, and finally a gene from Clostridium B. And all of these genes that would not normally be happening during this pathway have been engineered into the pathway of the E. coli. So now the E. coli is able to produce isopropanol. So when we're talking about production of our desired product, we also want to make sure that we're not producing too much of any product that we like don't really want to be having. So if you're going to look at this chart on the right, most of these yields for isopropanol are pretty good. Obviously, we want to have the one with the highest yield, so that's 4. But if you look at chart 5, this is where we start to have a real problem. So isopropanol is produced in this, and it's obviously you would like isopropanol to be produced. That's the whole goal of engineering this E. coli. However, we wouldn't want to use this pathway on the far right because it produces a large proportion of acetone. And acetone isn't what we're after. Acetone is just a byproduct. So since this particular combination of engineered genes would produce a lot of acetone that we wouldn't be using, it's not a good choice for us to use that particular pathway. So in this case, we can see that the pathway that we would most like to use is pathway 4.
Okay, so I'm going to go and talk about 2 more applications of industrial biotech that are a little bit more applicable to our daily lives and interesting to learn about. Okay, so next slide. So first up is Impossible Foods. I'm sure most of you have heard about the Impossible Burger, which is a plant-based meat alternative. The difference between this burger and a lot of other veggie burgers or plant-based burgers is that this burger uses tools of synthetic biology and genetic engineering in order to produce a burger that very closely resembles real meat. The way to do this is by targeting the hemoglobin molecule that is found in a soybean root nodule. The image on the bottom is showing us a soybean root nodule, and the reddish pinkish hue is the presence of the hemoglobin molecule. A form of the hemoglobin molecule is present in every plant and animal as it's an iron-containing molecule. Hemoglobin is an oxygen carrier in heme protein found in nitrogen-fixing root nodules of legumes such as soybean plants. Scientists discovered that the extraction of this hemoglobin in these soybean root nodules closely resembles the heme molecule found in blood and meat in animals, which translates to that meaty flavor in steaks or burgers. Therefore, scientists decided to use this molecule derived from a plant and create vast amount of the heme molecule that will be included in the meatless burger to give it that meat flavor and taste. Heme is essentially what makes meat taste like meat. At Impossible Foods, their plant-based heme is made via fermentation of genetically engineered yeast. They start by extracting heme from the root nodules of the soybean plants and analyzing that DNA for the production of this molecule. Then they chemically created and inserted these genes into genetically engineered yeast that use fermentation processes, earlier explained, in order to produce heme in vast amounts. This application of synthetic biology to produce a plant-based alternative to the classic meat burger has potential to greatly impact and benefit our environment. Impossible burgers generate 87% less greenhouse gases. They require 95% less land use and 75% less water to produce these burgers compared to a typical burger from a cow. Therefore, the adoption of meatless alternatives such as the Impossible Burger that closely resemble a meat burger can help reduce negative impacts and environmental damage of the meat industry. Next up, I'm going to talk about, and lastly, I'm going to talk about the textile industry and specifically a company called Bolt Threads. The manufacturing of textiles is actually the second largest source of pollution on the planet. Therefore, companies like Bolt Threads aim to reduce the environmental impact of the textile industry. Many of these companies are smaller as of now, but they're interesting to learn about in order to spark your own imagination or curiosity within the field to be able to understand the limitless and possible impact that synthetic biology within the industrial biotechnology field can have on our own environment and within industries such as food production and textiles. Bolt Threads manufactures something called microsilk. Microsilk is a silk-like biosynthetic fiber that is made through the process of simply fermenting water, yeast, and sugar with spider DNA. Bolt Threads are essentially, Bolt Thread is the company's essentially making spider silk without actually using any spiders. Spider silk has properties that include high tensile strength, elasticity, durability, and softness. Bolt Threads began by studying spider silk proteins spun by spiders and determined what gave the silk their incredible properties. The spider silk DNA, or the spider DNA, was then genetically engineered into the yeast cells, and through fermentation, a liquid silk protein is produced, which has a very similar look and texture as glue. It is in the same natural state as the liquid protein that actual spiders extrude from their glands and then form into fiber. So therefore, this biosynthetically made liquid silk protein is extracted and then spun into fibers the same way as common industrial fabrics such as acrylic or rayon are made. This company is an example that represents a lot of other companies that are doing similar things in the field of textiles and food production and biofuels and so on. All of these companies are looking for ways to implement tools of synthetic biology and genetic engineering in order to create biosynthetic alternatives of common industrial products that tend to produce a lot of waste and have negative environmental impacts. These alternatives that we're seeing within these industries is really the future of industrial biotechnology and can hopefully lead to a greater overall impact on current major industries as well as our environment.
Right. So, as just a little bit of a summary of what we've done, industrial biotechnology is the exploitation of enzymes, microorganisms, and plants to produce commercial products. Synthetic biology can be used to improve industrial biotechnology through engineering of microorganisms to create new or different products and/or improve metabolic processes that increase product yield. Industrial biotech has many applications across different markets with many different products, as can be seen with the diversity of the examples that we've presented today. It can be used for pharmaceuticals, for textiles, for fuels, for foods, even for plastics. Many different things can come out of this process. And it has the potential to develop industrial processes that are both cost effective and sustainable. All right. So that's it for today. I hope you enjoyed our first virtual lesson. I realized that it's a bit shorter than the ones that we normally do. It's because we aren't able to do our interactive portions like we normally do. And I realized that we covered a lot of information and we weren't able to pause to see if anybody had questions. So please, if anybody does have questions, feel free to contact the Systems Bio email. I'll be checking it really frequently over the next few days as people start to watch and process the information that we've given. And I'll do my best to respond to everything in a timely manner. Also, I'll be sending out a survey to see how people feel about this new setup, if we think that we need to do something differently, if the quality of audio and video on your computer is working okay. That survey will be coming out to you today, so please fill it out for us. Other than that, everybody, I hope you're staying safe and healthy. Next week, we'll have our next video posted, and that will be talking about principles of biophysics. Great. Thanks, everybody.
Thank you, guys.
Bye.